Good morning, everybody.
Morning.
Sorry for being a touch late. I actually got distracted reading about Mendelian genetics. How about that? You may or may not believe me, but it's actually really true. And also taking the dog out as well. Right, so what do we have to do today? Today we have a few minutes if anybody has questions to ask about material we covered last Thursday. This is something that's going to be particularly important for this class, this section, right, Tuesday, Thursday, because you're going to be taking in a lot of information in one go rather than the kind of more steady drip of the Monday, Wednesday, Friday section. So I'm going to sip my coffee for a little bit. If anybody has any questions, fire away. Oh, that was really good coffee. HEB. Lydia, you unmute. You don't have to raise your hand. You can type it out, whatever you want to do.
Can you hear me? Okay. I was curious about last week we were talking a lot about resistance and how you can develop resistance. Is that how genetic is that for humans when that happens?
In terms of chemotherapeutics?
Well, like my dad, he's diabetic, and he's recently, I mean, he's been diabetic for 30 years, and he's been taking insulin, injecting
Right.
it. And only recently he's just become really resistant to it. So his blood sugars have been like 300 and stuff.
That's a really interesting question. Well, obviously the mechanism is going to be different depending on the context. In terms of insulin, it's actually insulin resistance is one of the things that leads to diabetes. Hang on, Lindsay, I'll get to you in a question. I'll just finish answering this question. So essentially, well, I'm not an expert on diabetes because there's more types than I know of. I'm sure of that. But the diet-based diabetes is typically arises because the continual high levels of insulin in the blood, right, which are the result of your blood sugar levels continually being above what the band that is safe or desired, right, in terms of homeostasis. And so I'm not sure the exact mechanism, but essentially what's happening is that the receptors for that insulin, which are on your typically your muscle cells, your liver cells, things like that. So those are the cells typically that or tissues that are responsive to insulin. And if they're constantly being exposed to insulin, over time they are less likely to respond. I don't know the exact mechanism for that. One idea may be that the receptors kind of get gunked up, right? It's kind of like a non-science term, you know, so they don't actually, insulin is unable to bind or elicit response. Another is that those receptors are being removed from the cell membrane quicker than they can be replaced. And so the actual ability to respond to insulin decreases over time. It's actually, it's kind of weird because I had a student in my class last semester, proper semester, and he's a type 1 diabetic and he's having to change insulin types because the type that he was on is no longer working. And why that might be, I've absolutely no idea. Something due to the precise shape of the insulin molecule and how it interacts with the receptor would be my guess. Chemotherapeutics are different. The resistance there is more similar to antibiotic resistance in bacteria, which comes through well, typically in bacteria, there are three ways you can have antibiotic resistance. One, the target changes, so that whatever that chemical was binding to now changes in shape somehow, so it's no longer recognized. The other one is that the cells pump out the chemical faster than it can go in, right? So essentially it's not actually present there anymore to be able to do that. And the third is that the cells actively break down those chemicals via enzymes. So that could be similar to what we were talking about in terms of how you respond to pain mechanism, right? Inherent changes in your genetic code, which affect your ability to metabolize different compounds. So really, resistance is a bit of a catch-all term, to be honest. So it's kind of, in a way, it's almost sloppy to say resistance, because really you could mean a lot of different things. Does that help answer your question?
Yes, sir. I also wanted to ask about PCR. You were talking about how that kind of like changed everything. What was the discovery of that like, and when was it?
Okay. You only get that's your last question, right? Because I've got other ones on the chat board that I need to answer. So that came from our knowledge of how DNA replication occurs in bacteria, typically. So we were able to figure that out because bacteria are fairly simple, and essentially, you may not believe this, right? But the original PCR was done by using 3 different water baths. And you'd actually have to sit there. Well, not me, thankfully. This was gone by the time I had to do it, because I would have been bored off my socks. There wasn't even internet like smartphones at that time, right? So you had to put your tubes in the denaturing water bath, like at 95 degrees, and then you moved them to the primer annealing water bath at whatever degrees, and then the polymerase extension water bath. And so really that's just using the components that are present in DNA replication. So the 2 real changes that changed that was, one, the discovery of hyperthermophiles in the Yellowstone National Park. If anyone's been there, it's just an amazing place, really is. And essentially that allowed scientists to use DNA polymerase that operated at a temperature higher than the one at which the primers bound to the DNA. And so that allowed PCR to be considerably more specific. Before that, it was very almost semi-random what things would get amplified. The second big change was the invention of the Peltier cooler. So I don't know actually how many of you, we've got one in the back of our car, those little 12-volt mini fridges that you can put in your car, right? And they have a little fan going on, and they keep your bottles of water cold for Texan heat and stuff. Well, they have like a little kind of little heat sink in there, and it's able to, you basically pass electricity through it, and it causes a temperature difference on one side versus the other of the heat sink. And so essentially, whenever you use a PCR machine, you have one of those in there, and it's able to change the temperature very, very quickly. And so that did away with the putting things from one bath into the other, and that sped things up enormously. So those are the 2 real big things that changed PCR and molecular biology. All right. Sorry, going to Lindsay's questions. The being able to see the homework questions after you've submitted it. So I have it set, and I can go back in and check this because every semester I seem to have problems with Blackboard. I have it set so you can see the answers after the due date. You won't be able to see them after you submit the homework, but as soon as the due date passes, like the following day, they should be visible to you. So if you don't see that, I'll check after this, get my trusty rusty pen and hand out. This worked really well last time, actually. Because sometimes the options don't carry over when I think they do. Sometimes Blackboard is just a pile of junk. And I'll double check that. But go back, if you haven't checked it since the due date has passed, Lindsay, go back and check and see if you can see the answers. Hopefully you should be able to. I'm just gonna respond to Valentino's, well, this is a private question, so I'll answer that privately. Question from, I'm just going to skip Cross just a second because Catherine's question is on a similar vein about the questions of the homework. We will most definitely be going over chi-square stuff and crosses like you would not believe. In fact, you may come to regret asking that question. Yeah, you'll be sick and tired of chi-squared tests and crosses and things by the time we're finished. Don't worry. We'll definitely be going over that. So sometimes the homeworks and the lecture material and the labs they're kind of like wacky races, right? They don't always perfectly line up. So sometimes you'll see stuff in the homeworks which are a little bit ahead, sometimes a little bit behind the stuff in the lectures, just by the nature of them, really. Question from Cross. This is an interesting one about epigenetics that might be passed down from parent to offspring. So this is a very difficult question to answer because it's a very difficult thing to study. So there are two good studies on epigenetics, because obviously we're not allowed to study, like, experiment on humans. There's a bit of an ethical no-no. But the two studies are for smoking and kind of metabolic diseases like heart disease, diabetes, things like that. So the first, smoking, that was done in Sweden, I think. And I'm not very familiar with that study, so I can't really talk much about that one. I think it basically showed if you smoke, then your offspring are more likely to smoke, but I'd need to check that. The second one was done on or using data from people who were pregnant during what was called the Hunger Winter during the Second World War, which is when the German forces essentially imposed a blockade on parts of the Netherlands, which was 1944, I think. I can't remember. Basically after the attack on Arnhem, the bridges across the various rivers in the Netherlands. Watch A Bridge Too Far. It's a really cool film if you like war films. Anyway, so essentially they looked at individuals who were in utero before that famine, and by famine it was essentially about 400 to 500 calories per day per person. Right, so no, that's essentially starvation rations. And it lasted for about 4 months, 5 months, something like that. And so they looked at people who are in utero, so essentially in their mother's bellies before, during, and after that famine, and then followed them and their offspring through the kind of birth records database, which is pretty thorough in Europe. And what they found was that essentially, if your grandmother was pregnant with your mother during the famine, then you, as an individual, were more likely to have heart disease or diabetes or some other things that were bad for you in terms of diet. So that's essentially looking at people who were pregnant, what's that, like 70 years ago? Yeah, 75 years ago, something like that. And so those traits, epigenetic traits, were potentially passed down through those different generations. And part of the crazy thing about that, and this is something that I love talking about, is that if you are a woman, which I'm not, obviously, but if you're a woman and you're pregnant with a daughter, then towards the end of your pregnancy, I think it's in the 3rd trimester, essentially your daughter who's in your uterus has already made all of the eggs that she will have for the rest of her life, the next generation. So really, you have 3 generations in one person, which is a really, really crazy thing to think about, right? So essentially anything that the grandmother experiences can potentially be experienced genetically by the third generation, which is, it's just a kind of a mind-blowing thing. Let me just quickly answer a question. Oh, no.
Yeah, I
All
don't know who was asking about the PCR, but if you watch Forensic Files, like, they go into that a lot because
right.
up until like the late '90s, they were only using blood type when they found blood at that scene.
Uh-huh.
Until they started, and then they started using PCR, and they're like, "Oh yeah, you know what? It actually was this guy, not just his blood type."
Yeah, it's quite astonishing how quickly things have moved on,
Yeah.
and how specific things can be. I can't remember it specifically. I was reading something in the news about how recent advances in being able to extract DNA from samples and surfaces. So the amount of DNA that you need, which used to be quite a lot, right?
Mhm.
So you used to have lots of skin under fingernails or something. Now they can actually get it from one or two hairs.
Yeah.
And they've actually gone back and reopened a lot of cold cases and have been able to actually secure convictions from, murders and the like that were, carried out 20 or 30 years ago. Actually, wasn't that's how they caught the Golden State Killer?
Mhm.
They built a genetic tree, and from, I'm not sure entirely what, like 23andMe, I think, some kind of, send out your spit and get your genetic stuff back
Mhm.
service, and basically snuck around and snagged some hairs out of his trash, and that's how they got him,
I
which
mean,
is
you gotta do what you gotta do.
pretty amazing. Let's see. So, so talking about, we're not going to go too much into epigenetics now because it's a rabbit hole that we would not emerge until it's kind of time to have lab. To be honest, it's a very interesting and complex question. So in terms of like single one-off events and epigenetics, so the example was a car accident, for example. The answer is probably no, because if you think about the context in epigenetics, really think about timescales, right? So in terms of natural selection and evolution, like acting on DNA sequences, we're talking about timescales of, you know, probably in the range of several hundred years to several thousand years, right? So these are, and think of, changes like glaciation or, increase in temperatures or things like that, which happen over, multi-hundred years or thousands of years. That's what will affect the DNA sequence of a population, right? Allele frequencies, essentially. Now that can't change quickly, right? Because each generation for humans is like 20 to 25 years, give or take, right? So there are events that occur in much smaller time frames and generational time frames, like famine, really high levels of stress. So if you're growing up in the middle of a civil war, for example, right? So imagine the children that are growing up now in Syria, or have been growing up for the last, Jesus, is it like 10 years that it's been going on now? 8 years, something like that. Almost certainly their epigenetic marks will be changed, or the marks that they inherited from their parents during pregnancy will be different, right? And so essentially what epigenetics is all about is preparing your offspring for severe but short-term changes in circumstance. Droughts may last for, 4, 5, 10, maybe 20 years at most. So you want to, give your whatever pops out, you know, a better fighting chance to deal with that. But you don't want to change, you don't want the DNA sequence to change long term, because, you know, that's not a long term situation. So really, when you think about epigenetics and natural selection, you have to think about time, you know, and the context of whatever's happening at that time. So one-off events like that, no, they're not going to change what will be passed on to your children. But if you are under considerable stress for a long period of time, then yes, that most likely would. Right. And so think about that as well in terms of growing up in, you know, inner city Detroit versus one of the swanky suburbs way out on the outskirts of Detroit. The experience that you'll have there is going to be very different to other places. Right. So that's another thing that's really interesting. I don't want to get into kind of like racial politics, because that's not really where I want to go with this. But the circumstances in which you are gestated, in which you grow up, are different. And those differences, depending on their severity, will have consequences, right? And so, and those consequences are ones that you see over multiple generations. It's a pretty cool topic to think about. Anyway, I think we've been talking about fun stuff too much. We've got to talk about, well, not boring stuff, which is kind of the implication, but things that we actually need to study. So awesome questions though. So essentially what we've got to do today is talk about the last bit of the flow of genetic information PowerPoint, which I'll bring up in just a second. And then we need to, pardon me, get on to Mendelian genetics. It's all about peas. One day I'm going to make a really cool Mendelian genetics PowerPoint on cats. Actually, I think I did for GenBio 1. I should have dug that out and reused it for this one. Anyway, so I can't remember exactly where we got to last time. I think we were about here. Genetic code. Correct me if I'm wrong. I think that's about right. I'm going to try and bring up the chat box again. Come on.
Yeah, that I think you did leave off right here at the.
Okay, cool. I was pretty positive that's where it was. Let's see. I'm just trying to get the chat up. Oh, that's kind of worked. Honestly, it's like all these things seem to be made by people that don't actually use them. All right, let's get back again. Ah, fudge nuts. That's kind of gone and covered the chat. All right, if you have a chat question, just cough. Oh, there we go. Back up again. There we go. All right. So essentially, can't remember if I mentioned this or not. The benefit of this degeneracy, right? So, you know, these codons aren't like slouching around street corners and acting all kinds of suspicious. But degeneracy in this term means that you don't have to have all three bases of the codon correct for that amino acid to be encoded or incorporated into the peptide. So leucine is obviously and always a good example. It's one of, I think it's the only one. There's always one which is kind of like scattered around somewhere. Anyway, but leucine is encoded by 6 codons, right? So if a mutation were to occur in the third base, like the third base of the leucine codon, CCU, for example, was changed, then the amino acid that codon encodes would not change. So in a sense, this is like a buffer against mutation, right? Because mutation is random and there's a chance that it will occur in the coding region. And if it does, it reduces the likelihood that it's going to change the protein sequence, right? So it kind of gives you a bit of a, buffer.
Some leeway.
Go on.
Like 33% leeway if your body accidentally mutates or the code changes.
Correct. Yeah. And it's kind of interesting because when we think about mutation and evolution, we tend to think about the changes that benefit the individual, which have like some kind of fitness benefit, whatever that might be, you know, being able to run faster, walking upright, you know, for cooling brains and things like that. But the vast majority of mutations are not good for you. They're either neutral. Well, vast majority are neutral because the vast majority of our DNA doesn't code for proteins. But of the mutations that do occur in coding regions, most of them are bad for you, generally speaking. That's why we have such a large panoply of genetic diseases, right? And very few superhumans. I'm sure there's some. I'm sure Elon Musk must be one, right? I can't stand that guy personally, but
No.
anyway, the thing is a complete and total jerk. But in terms of why? Oh boy, I think I'll add that to my list of rants, along with giant pandas and a few assorted other things. I'm not going to get into that. We've spent too much time on tangents already. Another time. Yeah, don't get me started, Gabriella. Anyway, all right, I'm going to close this chat unless you stop trying to distract me. And so essentially, this is like the Rosetta Stone between mRNA sequence and protein sequence or amino acid sequence, right? And the bits, right, that actually do the translating, right? You might think that the ribosome does, and actually you'd be wrong. The ribosome does the making of the protein, but it's the tRNAs, the transfer RNAs, which actually do the translating, right? Because they, through another enzyme which actually sticks the amino acid onto the end of them called, what the hell is it called? Amino acyl transferase, I think, something like that. And the anticodon, right, which is the sequence you see at the end of the tRNA, that's what kind of translates between the codons in the mRNA and the amino acids in the nascent polypeptide that's being made. The ribosome is really a kind of structural edifice in protein form, and the ribosomal RNA is the enzyme that sticks these things together and removes them from their tRNAs as well. So moving swiftly on, RNA is actually a super cool molecule. You might not realize this. If you don't, that's totally fine. But just in case you, come on, get you back. Oh, I hit the chat by accident. Never mind. It's actually the molecule which there's several competing theories as to the origin of life, right? But there's actually a very large kind of movement behind something called the RNA world, right? Because RNA is the only molecule that we know of currently that is in current existence that is both a store of information like DNA is, but can also act as an enzyme like proteins can, right? So in theory, RNA or whatever the similar kind of molecule that was around at the beginning of life, right? So a long time ago, 4 billion years ago or so, it maybe looked something like RNA, right? And RNA is particularly funky because it can bond with itself. So it can base pair with itself just the same as DNA does. But rather than forming like a rigid double helix, it can form a really diverse array of structures, right? These kind of loops, which you see down here. It can base pair with itself to form stems, right? Where it's essentially double-stranded RNA for a stretch. And when you look at these images, this is a real funky way of drawing a tRNA. But essentially, it shows you that there is a whole bunch of structures in tRNAs, right? Which essentially allow it to do its job.
Excuse me.
Go on.
Sorry to interrupt you, but just real quick. So if the tRNA is bonding, does that also make a protein? I thought it was, the half of the DNA with the mRNA that would make a protein.
So kind of to unpick that a little bit, as you go back up here. So no, not at all. This is a very temporary DNA RNA hybrid, right? So it only really exists during the process of transcription, right? The rest of the mRNA is kind of dangling off in the breeze, essentially, kind of that's already been made down here. This mRNA is then via a bunch of other things translated into protein, right? So the DNA, let's think of that kind of up here, the mRNA and the protein are all distinct molecules, right? The point about RNAs, not necessarily mRNA, although that can form structures too, but RNAs in general is that they can form structures of their own by bonding with themselves, right? So going from, it'd be easier to just draw it. Let's see. Yeah, trusty rusty whiteboard. You know, I should just use my eraser. That'd be a lot easier, wouldn't it? So essentially, this is like just being in an office, right? Rather than sitting on my couch. So let's just call this an RNA of some sort. I'm not sure what, doesn't really matter.
Okay.
But essentially that's going to have regions within it which are complementary to each other. Right? And by complementary, this might have an A here and a U here. Right? So when this RNA molecule is folded over, that A and that U will be able to base pair just as an A and a T would in DNA. And so after a while, what you'll get is something that looks potentially like that, right? So this is, what would that be? 1, 2, 3, 4, right? You'd go from a linear molecule like so at the top, and you'd end up with this, you know, 2 to 3 dimensional molecule like this at the bottom, because these regions here, there's 2 and 3 regions, will base pair to form double-stranded RNA. The 1 and the 4 won't, will, but they'll form a region down here. And then all the rest in between, oh, my dog's being a jerk and chasing my cat. The ones in between will form these loop structures because they're not complementary to each other. Right, and so by forming these structures, essentially they're able to, it's going to make that a little bit bigger so you can see it a little bit more easily. These are able to form 3-dimensional structures and have catalytic regions. They can have regions that are able to promote enzymatic reactions. Right, and so this isn't a protein, right? It's still just RNA, but it acts as if it were a protein in doing something. So it's now able to catalyze reactions. And this, if it's catalytic RNA, that is called a ribozyme. Hopefully you can read that. I've got a really shockingly bad handwriting. Right.
Yeah, I can read it. Thank you.
And an example of that is ribosomal RNA. So the RNA that actually catalyzes peptide bond formation in translation is not the protein in the ribosome. It's the ribosomal RNA, which is a really weird thing to think about, you know. So, because one would assume that it's the protein part of the ribosome, and no, it's not.
True. Thank you.
You're welcome. Yeah. So, question from Aisha. Those The regions of the RNA molecule that are double-stranded because they're paired together, those are called stems. And the bits that do not match each other, those are called loops. You know, not very imaginative names, unfortunately. But yeah, that's just what they're called.
That's not the worst thing.
Honestly, like look at any gene names that Drosophila scientists called things, and you'll be blown away. There's one at a conference I went to many years ago where this guy was talking about a gene that he'd called shaven baby.
What?
It's really weird, like listening to a whole like 15-minute research talk with this guy constantly going, and shaven baby does such and such, and shaven baby this, and shaven baby that. It's like, you guys are nuts. Had a really good long argument with him about gene naming conventions after the after the talk. Yeah, well, the Drosophila people are like, oh, you C. elegans people are so boring. You know, you only have like 3-letter gene names, and we have real fancy ones, and you know, so on and so forth.
Yeah, how many people can remember our genes, though? Oh, that's right.
Yeah, yeah, exactly. That's true. I mean, there's actually one called Sonic the Hedgehog, for example, which is a really important one in growth and development. And the theory goes is that it was because his kids were playing Sonic the Hedgehog on, I think it was a Sega console back in the day. Yeah. And he was like, oh, that mutant embryo looks just like Sonic the Hedgehog. And so that's what he called it.
Interesting.
Oh, brother.
Yeah. Anyway, so just to finish this PowerPoint up, one thing to really get across, and I don't think we'll be talking hugely about mutations, maybe a little bit about them later. But essentially, mutation is the only form of novelty in terms of genetics. Now, epigenetic marks, kind of, but those are not durable. They tend to disappear after a couple of generations. So mutations are really the only truly, you know, multi-generational heritable change in genetic material, right? And they occur typically via DNA replication is one mechanism, right? So the inherent errors in copying DNA every cell division, they can be caused by other genetic elements called transposons, which jump around and cause all kinds of damage. And they can be caused by what I like to call external insults.
What did you call that? Transposons?
Transposons. Let me write it out in the chat. There's a whole PowerPoint on them, which you'll be. They're called jumping genes. If I can spell genes right. God, that's embarrassing. Geneticist. And they were discovered by a very, very cool scientist called Barbara McClintock, who got the Nobel Prize for her discovery. And she discovered them in maize, I think, maize or corn, one of the two. And she actually had to fight for a long, long time to get anybody to believe her. I don't think it was necessarily because she was a woman, although that may have been part of it. But part of it is because no one basically just believed her that you can have mobile genetic elements. Yeah, so anyway, and they make up a huge chunk of our genome, essentially. They're like kind of ancestral cruft in genetic terms. But anyway, we'll be talking about those a lot later on. They're pretty interesting things to look into. So generally speaking, mutations are either neutral. Neutral means that they don't have any apparent change in either a trait or the fitness of an individual, right? So if a mutation occurred in a long stretch of repetitive DNA that doesn't do anything, that we know of at least, then it's not going to have any effect on the fitness of that organism. Now, if that mutation, and these are really important to stress, with the exception of transposons, and that's kind of random, mutation is random, right? So there's nothing that, until recently, with advances in biotechnology and transgenics, there's nothing that we as organisms can do to change our own genetic sequence. Any changes that do occur are simply random, right? And so if you, neck a container of 5-bromouracil, right, which is generally a bad idea, those changes, or you go swimming in a spent fuel rod pool, nuclear reactor, the changes that you will receive in genetic terms will be totally random, right? And that's also one of the reasons behind, Uncle Joe's like 106 and smokes a pack of 20 fags a day, right? Totally fine. And then other people get lung cancer in their 40s, right? It's just random. And there's other things to that as well. But, and so mutation is both random and generally either does nothing for you or does something bad. There are very occasionally mutations which enhance your fitness, right? And so by fitness, we mean the ability for you to survive to reproductive age and reproduce. That's generally what we mean by fitness. And obviously there's lots of different ways of thinking about that. Being able to outrun the saber-toothed tiger is a enhancement in your fitness because you're not going to get eaten, which means you can reproduce. Being able to metabolize lactose, right, is an enhancement to your fitness because it means you can drink milk, which opens up other sources of nutrition, right, which will again make it more likely that you'll survive to reproductive age and reproduce successfully. So it's really that. Go on.
Do mutations like have a lot to do with like Darwin's way of thinking?
Well, Darwin was really the first to think about the inherent differences in us, in how some become more prevalent in populations. So his original work was, or theory was really descent with modifications, which was really that your offspring and your offspring's offspring, and essentially your lineage, you have the genetic information of your ancestors, but that is modified based on your personal circumstances, right? And so what he was really describing, even though it wasn't couched in those terms at the time, was natural selection, right? And so really, and this is kind of I didn't do this, but it's a pretty useful image. Natural selection is the funnel which both gets rid of bad mutations, right, deleterious mutations, and increases the frequency of beneficial ones, right? And so essentially mutations, what they just lead to genetic diversity, right? So our genetic diversity as a group of individuals or as a city or as a country is essentially the result of all the different mutations that all of our different varying ancestors accumulated over the last 50,000 years or so, right? That's why we're different, and that's why we're also largely the same, because there aren't really that many differences between us. We're, I don't know, 99.999999% identical genetically, but obviously the small changes that we do have make a fair difference in terms of what we look like externally. But natural selection, and this is really what Darwin was kind of fumbling around at, even though he didn't realize at the time. And it was a very revolutionary theory at the time, because there were many people actually thought, and this is kind of tinged with the brand of Christianity at the time as well, was that man was perfect and fixed, right? So essentially we were created in the image of God, and therefore we could never change, because that would imply that God was not perfect, right? Which is obviously not something that fits within the Christian canon, or really any kind of religious canon. And so what he was suggesting at the time is that essentially what was created, even though he obviously did believe that it was created by God, right? Because he was very much a Christian man and a believer in God. Whatever God originally made has been changed by natural forces over time, right? And that was, I mean, if he'd tried out selling that, a few hundred years earlier, he probably would have been like nailed inside a box and thrown off a ship, or burnt at the stake, or buried under stones or something like that. And so really what he was talking about is this process here, this funnel. And really natural selection is a external pressure, right? Whatever that may be. In his finches, it was drought, right? And food availability and seed size, right? He focused on finch beak size, which correlates with the type of seeds that are available for those finches to eat. And so over time, depending on that pressure and how strong it is, right? If there is a mutation in this kind of general pool up here, which increases fitness, then as a result of that environmental pressure, the frequency of that allele will increase in that population. So in the case of finches, during a drought period, seeds get a lot harder. Their shells become thicker, or whatever you call the bit around the outside of a seed. And so finches need bigger chompers to crack those seeds, essentially. And so if there is a mutation, random, again, right, this isn't directed in any way, but just a naturally occurring mutation that either occurs or is present in that population, then that increase in seed size will favor that mutation if it increases the beak size of the finch. And so over time, essentially, that trait becomes more and more fixed within that population. And if there happens to be another population of finches kind of like over there, which is like, ooh, you big beaked finches are bloody ugly, I'm not going to reproduce with you, right? Then essentially you start seeing that bifurcation of populations, which potentially leads to speciation. Oh, dang it. Sorry, I had a private. Thank you, Valentino. I appreciate that.
I have a question for you.
Go on.
So it kind of dabbles in evolution and mutation, but I mean, we've had smog in the cities, since basically the Industrial Revolution, which could date back to like 1720s roughly.
Mm-hmm.
You think
pea soupers, they used to be called in London. I don't know if you've ever had pea soup. My mum used to make it for some reason. Bloody disgusting. But it's like basically you get a whole load of peas, dried peas, and you boil them for like, I don't know, days, and you kind of like blend them up, and that's pea soup. And so back in the day when they used to have coal-fired power stations in London, they used to have fogs or smog, which they used to call pea soupers, because it was as thick as pea soup. There you go. You couldn't actually see your hand in front of your face.
Oh, that is.
Yeah, it's pretty insane. But anyway, sorry, that was just a
no,
cultural aside.
Yeah. But my question is, do you think that our bodies are slowly going to be adapting to the smog, at least those who are in the cities? Because, I mean, you're talking, what, almost 300 years now? We've been exposed to smog. Do you think our lungs would adapt to...
Potentially. So there's a lot of different things that play into that, and I'm not really enough of an evolutionary biologist to be able to answer it properly. But if those populations, if that pressure remained and that pressure reduced fitness, which we know that it does through asthma and various other kind of lung diseases and stunted growth and things like that. And this is the key, that population did not move, right? So if there's little population flow or migration, then you would see change over time. Whatever that change may be, I don't actually know. Maybe people end up getting shorter or something, who knows what. But the problem with kind of our current day situation is that even though there are selective pressures, there's so much population flow and migration and admixing that it would be hard to see what effect that has. So even though I'm sure that that does occur, being able to tease it out from all of the background noise and the various other stuff that's going on in terms of gene flow would be really difficult. But one thing that is actually kind of interesting is when you look at onset of puberty, right, particularly in girls, that has been getting younger and younger and younger. And part of the reason for that may be due to the presence of contraceptive hormones in the water supply, right? So even though they're very, very low levels, that may actually be changing some aspect of our biology as a result. What the consequence of that is, I've absolutely no idea, because yeah, that's actually another idea too, Marina. Yeah, I mean, that kind of gets us to how complex. So Marina said that might be due to body fat content as well, which is also true because our diets have changed considerably over the last 20 to 30 years, if not a little bit longer than that, maybe back to the 60s probably. So yeah, but that's also a really good example of how difficult it is to answer those kind of questions, right? Because, you know, why are people changing the way they are? Well, we're very complex organisms and we live in complex environments. That's why a lot of the kind of real breakthrough stuff which leads to our understanding in terms of evolution happens to be either in simple organisms or in organisms that exist in simple environments. So the Galapagos Islands are a great example of that.
Yeah.
You know, there's very little gene flow because they're islands, right? And they're also islands a long way from anywhere else. I actually nearly went there once. I traveled through South America when I was a wee young man. In my, I think I was 20. Yeah, I had my 21st birthday in, what was it, my 20th birthday in South America? I can't remember. But I almost went to the Galapagos Islands, but I just kind of balked at the cost. It was about $1,500 at the time. So I didn't. Kind of regret that a little bit, but you know, is what it is. One day maybe. Anyway, just to finish this stuff off, let's make sure I'm having more questions. One can also categorize mutations in lots of different ways too. And it really depends on context. And so that's something that I really kind of get back to a lot. Context is everything. Whatever you're talking about in terms of biology, right? So, you know, the context of what Darwin was working on was very much shaped by the environment that he was working in. If he had tried looking at stuff like that in, I don't know, industrial Britain, he may have come to other conclusions, for example. And so in terms of mutations, kind of the consequence of those really depends on context, right? It depends on where those mutations are, right? So if you, like I said, I think, maybe this was the other section, any mutations that you accrue as an individual, or even me, as I'm essentially post-reproductive age, at least one hopes, but any mutations that you accrue in your somatic tissue, which is essentially everything that's not going to result in the next generation, die with you, right? So you may have like an amazing mutation occur, which gives you like massive muscles or something. It doesn't matter if it's not in your germline, right? Because essentially as soon as you don't exist anymore, that mutation dies with you, right? So however, if that mutation occurred in your germline, if sperm or eggs, then that potentially, and that's also very much potentially, right, because reproduction is random, could get passed on to your offspring, right? And so I don't know if I mentioned it to you lot or the other lot. When I teach two sections of the same thing, it's kind of a little confusing sometimes because I say one thing to one group and I forget whether or not I've said it to another one. But even say, actually, I'll use myself as an example. Say I had a mutation that occurred naturally for whatever reason in one of my sperm cells, right? And I was to, produce an offspring, and that mutation carried on into that offspring. That mutation, that allele, right, that change in a gene, let's say, would be present at one copy in, 1.2 million individuals in San Antonio, give or take, right? So even though that mutation may be like the best thing since sliced bread, its frequency in the population will be infinitesimal, right? And so it would require that mutation to confer an advantage to my offspring and get passed on to his or her offspring, and so on and so on and so on, for multiple generations before you would ever even be able to detect it as an allele in San Antonio, right? So if you think, it's actually a really weird thing to think about, right? Because we see the end result of natural selection, right? And that end result is potentially after hundreds or thousands of years. But at the very beginning, there was only one. Who gets that reference? Yeah, Kevin does. I think that was Kevin laughing. That's the Highlander, by the way, if you haven't seen that film. There can be only one. Rawr. Anyway, it also matters where that mutation occurs, right? If it occurs in non-coding DNA, then it may have no effect at all. If it occurs in a coding region, it could have a major effect.
Top off my coffee.
Oh, okay. Sorry, you're hidden by my chat box, so I was just checking to see if you were asking a question. If it occurs in the coding region, it can have very different effects depending on what it does, right? So if it occurs in essentially, the third base of a codon in, say, leucine, right, it would be a synonymous or same mutation, right? It wouldn't have any effect at all, right? If it resulted in a stop codon being incorporated, which would be what's called a nonsense mutation, it would truncate the protein, and that would be what would call a loss of function, kind of up here, mutation, right? It would eliminate normal function. So whatever that protein did, it wouldn't do it anymore, right? But also that depends on, do you have another functioning copy of that gene on the other chromosome? This is where we kind of start getting into Mendelian genetics, right? So whether or not you see the consequence of that mutation also depends on what your genotype is for that particular mutation, right? Which isn't something that's actually included in here. That's more Mendelian genetics. So I don't want you to stress too much over these words, right? But these are concepts we'll be hitting again later on in the course. So it's worth, at least familiarizing yourself with some of the language in there, right? Insertion and deletion, for example, right? What are those? They're actually called indels because we're sloppy and we like sticking words together. So that's essentially this PowerPoint done. I don't really have much more to say about this. Does anybody have any questions before I move on? And then we'll get started on the Mendel PowerPoint. Yum, yum. This is HEB creme brulee coffee.
Oh, how is that?
It's good. Sometimes I like diluting it a little bit because the creme brulee-ness can be a little bit too much, depending on your personal proclivities. But yeah, I like it. HEB does really good coffee. actually. I've never been unhappy with coffee that I've gotten from HEB.
I get mine from Sprouts. Their organic coffee is $10 a pound, and it's the same price as non-organic coffee. So I was like, yeah,
No, that's not bad.
yeah.
Yeah, if I was kind of not out in the burbs and kind of lazy, I would definitely be checking out some of the coffee shops around town.
Yeah.
But yeah, it's just a bit too much for an arse ache to try and get into the city, to be honest, from where I am.
Hmm.
Oh
What
yeah,
part of this town do you live in?
northwest, kind of between Helotes and SeaWorld.
Oh yeah, you're out there.
hmm It's a hell of a commute. My bike ride to work is 25 miles each day.
You bike to work every day?
I do about 9,000 miles a year.
Oh man.
Yeah, I have
And I
great
complain if I
legs.
have to park like 5 spots down in like the 3B spot.
Oh, okay. Well, I mean, that's good exercise, particularly in the middle of summer.
Mhm.
Yeah, so I haven't been riding my bike very much at all, actually, since this whole Corona thing. So I
I
need
do
to.
remember beginning class with you and having you like go on a tangent about your calves.
I know. I actually have had students comment on that. One of my students said, "You have the biggest calves I've ever seen." I was like, "Great, thanks." I think I'm going to take that as a compliment, even if it wasn't intended as such. Anyway, so because we've only got like 15 minutes or so left, and I need to start lab pretty punctually because I've got to get out of the house on time today. So I've got getting all my personal characteristics recorded by USCIS, which will be fun. So what I want to do is something a little bit boring, sorry, and just make sure we are all familiar with some of the terms and terminology in Mendelian genetics, right? Because when we talk about Mendelian genetics, crosses, different generations, different things, right? We use very specific words, and if you don't understand those words, you'll be lost. All right, so this is like another aspect of the language. Oh, and before I forget, I have tried Starbucks Colombia coffee. It's really nice. Nowhere near as good as the actual coffee in Colombia, which is just amazing. I actually started drinking coffee in South America when I was in Colombia when I was 19. Yeah, because my parents had terrible taste in coffee, and so I never drank coffee until I left university and went to South America. And I still can't drink my parents' coffee because they still have really terrible tasting coffee.
Yeah, mine drink that Folgers. No thanks.
Oh,
'Cause I
well,
drink
that's
I drink
actually
it black, so like I have to pour a buttload of sugar in there.
yeah. Folgers coffee would be delicious compared to the crap that my parents used to drink.
That's pretty bad.
That is pretty bad. Yeah, I mean they'd be quite happy drinking coffee that was like 10 years old.
No.
Anyway, so a lot of these terms, they're all in the PowerPoint anyway, that you can go over them again afterwards. Oh, sorry, Lindsay. Oh, is that my foot in my mouth? Sorry about that. Let me get that out of the way. Yeah, so a lot of these terms are ones that you're familiar with already, but they are important to know. One of the most important ones is this one. I can highlight it, right? We will be talking a lot about alleles, right? Alleles are simply genetic changes in a gene that differ from one individual to the next. That's it, right? There's no, most of the time we talk about alleles which change function, right? Particularly in like Mendelian genetics, things that make peas change color and stuff like that. But simply put, an allele is any genetic difference between a gene in one individual and another, right? There may be no effect on function as a result of that allele, right? So it could essentially be a neutral allele, which, right, does nothing very much. It could be something that improves function, like a gain of function allele. It could be a loss of function, it could be reduction of function, it could be whatever kind of function. It is simply a different form of a gene, right? And so we have, you know, millions of alleles. It depends on what gene you're looking at and what population you're looking at too, because the makeup of alleles in one population is going to differ from another depending on their evolutionary history, essentially. And also another one which may not be so familiar is that of a locus. That's not a locust, right? That's something that eats crops. This is the specific location of a gene on a chromosome. So it's like the physical map of where a gene is. It's like, it's just there, right? That's the locus. Right? So those two are ones you have to be familiar with. These are also ones which you should be familiar with already. You know, one hopes. The notion of homozygous, heterozygous, dominant, recessive, things like that. There are more types of traits than just these, right? But these are the only ones that we're really going to be talking about, in this class, right? If you take molecular biology, maybe, I'm not 100% sure, but you may be talking about other types of traits, which we won't be covering here. But essentially, you should already be pretty familiar with these. And one of the reasons for going through these ahead of time is that there are some pretty heavily baked in assumptions in Mendelian crosses, right? And monohybrid and dihybrid crosses are inviolate. They never change. They are always exactly the same. So what you get in one generation is going to be exactly the same for one monohybrid cross versus another. The genes may change, the alleles may change, the phenotypes, the traits, whatever may change. But what you get in each generation is always going to be the same, unless something funky is going on, right? So this is an important assumption for the parents. So in any monohybrid or dihybrid cross, we're not talking about test cross here, right? We're talking about monohybrid or dihybrid, and they are very specific things. The parents will always be true breeding, right? And so what that really means is what it sounds like it, right? If you have a dog, which is a, I don't know, a schnitzel or schnauzer or whatever they call it, if you mate that with another schnitzel or schnauzer, I think schnitzel is actually a type of steak in Germany, so maybe a schnauzer, you'll only ever get schnauzers, right? You won't get some like half funky looking mutt with really short legs, right? You'll only get schnauzers. Yeah, definitely schnauzer. Yeah, but schnitzel is really nice, actually. If you ever get a chance to try it, go to like Fredericksburg or something like that.
Love Fredericksburg.
Anyway, and so that's a really important assumption, because if that's not true, then you don't have a monohybrid or dihybrid cross, and B, you're going to get something very different out of your cross than if that were true, right? Hybrid is essentially, hang on, what's that say? Oh, interesting question, Priscilla. So if those dogs are purebreds, right, they're true breeding, and they mate with their siblings, then you'll get the same phenotype coming out, right? So you might have other issues, which is caused by inbreeding, but we're not really going to get into that. That's more of a another issue. But yeah, so if you are homozygous for a trait and you mate with another individual that is also homozygous, you'll only ever get homozygous individuals. You'll never get heterozygote. Mixed race child, well, that's a really difficult question, Marina, because we are far too complex genetically to be able to pass it down to simple stuff like monohybrid and dihybrid crosses. Actually, if you want a better example, go look up, what was his name again? The last Habsburg king of Spain. Shoot, I'll have to dig out his name. Here's a good example of a purebred. Actually, most royalty is, unfortunately. I mean, just look at King Charles. He's not a particularly good looker, right? But most royalty, because of inbreeding over many, many, many years and generations, is what can be called purebred. And I'll actually have to, I keep forgetting this guy's name, but he is an absolute doozy. Let's see if I can, oops. I think I just turned off my video by accident. Get out of it. I was trying to type something into Google. Oh, here we go. This one's a corker. Sorry, you got me off on a tangent. I got to stop letting you people do that. Right.
We don't mean to do it.
Yeah, right. This guy here, King Charles, there you go. King Charles II of Spain. He is what one could call a purebred, right? Because he is a result of lots of his aunts and uncles and nieces and nephews mating with each other. And essentially, he is the last, or was, because this is obviously some time ago, the last Habsburg king of Spain. And the reason why he was the last one is, well, just look at him. He didn't just look at, actually, it's really, really funny to read about. Like, there's supposedly like an autopsy of King Charles II, because he died when he was, 20 or something. He didn't actually speak, supposedly, until he was 6 or 7. Supposedly, he only had one testicle, and it was like a shriveled walnut. He was, well, anyway, there was lots of problems with him. So this is essentially what a purebred, true breeding human looks like. It's not a good thing. So when we talk about races mixing, essentially what you're talking about is mixing one group of alleles from one ancestral lineage with a different group of alleles from another ancestral lineage. And so in some ways that can be, I've got to get this guy off the page because it just causes me all kinds of disturbance looking at this guy.
Pretty hard to tell if it was a guy or a girl, to be perfectly honest.
Yeah, it's just not good. Oh, quickly from Marco, e-textbook, you should have an email from the library with your e-textbook. If you don't, you need to look at the Jaguar Day 1 info and get in contact with them. I don't have anything to do with that, unfortunately. But ever so quickly, just talking about going back to Marina's question, when you mix those different lineages together, you can have good things, which is like, I don't know, I mean, kind of getting into a little bit into the weeds here, but some individuals with mixed heritage are particularly stunning, right? Male and female, right? Is one way of thinking about it. Other ways, it can be really bad. Like if you, my dentist was always going on about this. He said he sees lots of tooth problems, in terms of needing braces and teeth removals in children of parents from different, not races, but different areas. Because, there might be a Caucasian, European tooth plan, palate, whatever you call it. And when that gets mixed with, say, someone from South America, like my wife, then you have a kid that needs braces, right? Because you essentially have this kind of collection of alleles kind of clashing or sets this particular thing, and then you have this one which causes problems. And it's an impossible question to answer, to be honest, because genetically speaking, we are so astonishingly complex. It's really, really difficult to answer any of those kinds of questions. And I've managed to tangent myself to the end of the class. And so there's only one slide left. Whew. And so the other thing that you also need to just refresh yourself with, this is essentially the structure of a Mendelian cross. Not a test cross, but a Mendelian cross, by which we mean a monohybrid or a dihybrid. And this will always be the case, right? So whenever we go through those, which will start on Tuesday, this is exactly the format that they will have, right? And it doesn't matter how many different traits you're looking at, they will always be the same, right? Assuming that you only have 2 alleles for each trait and a bunch of other assumptions in there, right? So if you're not 100% down with any of these terms, then kind of buff up on them. We'll be using them a bunch next week. And also have a gander through this PowerPoint, so you're just to brush up on stuff that you may have covered before but haven't looked at for a while. Let me just check the chat. I think someone had. Let's stop that. Go back here. Yeah, that's pretty much it, I think. Yeah, if you have questions on the homework, send me an email. If you can't access the, I've got it written on my hand. I'll check this after lab this afternoon, which means I can't wash my hand now. But I'll check to make sure that the correct settings are enabled to allow you to see your homework answers after the due date, because there's a chance that those weren't done properly. If you have any problems with the homeworks, obviously just send me an email and let me know, and we'll figure it out. But that's pretty much it. So I've got lab with some of you in about 12 minutes. So I'm going to run off, brush my teeth, stretch my legs, and then hey, I'm British. I've got to look after my teeth, right? Battling stereotypes since 1978.
I thought British never took care of their teeth.
What's that?
So I thought the stereotype was British never took care of their teeth.
We have better tooth care than Americans do, on average.
Oh.
It's on average, right? So some of us, so some Americans have amazing teeth, but some of them have absolutely terrible teeth as well. It's just Austin Powers. That's what I blame it on. 100%. We actually have a lot better teeth than people give us credit for. Anyway, I'm going to head off. I'll see some of you in 10 minutes, and others I'll see you next week. All right. So if I don't see you, have a great weekend, and catch you later.